Januaries are usually slow at work, so I'm currently working from home. I think I've been doing that since the start of January, and today is the 13th. And I'm going into the lab next week. So, what I'm doing is I'm using Asana to set up my tasks for next week. Because I work in the plant pathology department, another thing that I'm responsible for, or maybe the main thing that I'm responsible for, I'm the lead experimenter on the plant pathogen systems that we develop. So I'm just going through the microbial library of the fungus that we have on hand, and I created it. Quite a bit few of those images that you're seeing are from fungus that I personally have isolated. So why I'm going through it is because I'm going to develop model systems to work on. The second thing I do when I'm going to do my research, or I'm going to develop a model system after I've checked to see what pathogens, what fungus, and which plants we have at the lab, then I do a Google search or I search journal articles. So people who are maybe in second year of university, that's undergrad or grad school, you're familiar with literature searches when you go through peer-reviewed journal articles that other scientists they've written on the topic of interest. So I have my writing pad on the left, and then the article I go through. So this isn't a typical grad school research that I'm doing. So I'm not reading the entire article. I'm more interested in characteristics of the pathogen. How does it cause disease? What are the symptoms? What are the environmental conditions? What are the things that it needs in order to cause disease in the plants? How is it treated? And so three key parts of a pathogen system, or when you're thinking of disease, you think of the susceptible host. So that's why I have a disease triangle. And this is something that is key to plant pathology and other discipline. So it's a legit triangle. So I have the 3 points. It's dynamic, so it's not static. And in it, you'll have susceptible host, so the thing in which it will cause disease, it will infect or at the top or one end of the triangle. Then you have the virulent pathogens, so the thing that will cause disease, it should be able to cause disease, right? And then you have the conditions. So I'm just going through, I look at the methods to see what was done, because different pathogens, they require different nutrients to grow. And there are some nutrients that they grow best in. So some like a lot of sugar in their nutrients, in the food you're giving them. And then if I'm reading and I see a quote, which is really a citation that I'm interested in, then I'll go to the reference section, look to see who wrote that, and then I'll copy and paste it into my search bar so I can pick up that exact article and I can read it to see if they went into details about what was done. And I'll do that over and over until I feel like I have a broad cross section of information about my system, the plant growth conditions, what kind of lighting, what kind of nutrients, you know, um, what what conditions are needed for the disease. So the plant will have its own conditions, the pathogen will have its own conditions, but you also need specific conditions for the disease to show up in the plant. So things like a lot of rainfall, high humidity, cool or hot temperatures, and for how long. So because I use a growth chamber, so most of those conditions, I try to mimic them as if they were out in the field, out in nature, out in the wild. So I can't get like actual rainfall inside, but we'll have misting system that will stimulate rainfall. And so yeah, I'll highlight key points that I want to include in the document that I'm preparing. And so I'll prepare a document so that with written words, it will have images for others to use in discussion. So my manager will take this to upper management meetings and they'll have discussion on what we might want to work on because it has all the information to make those decisions. So it's like your portfolio, but a pathogen one. This is just a quick overview of what one of those pathogen portfolios or profile looks like. So as I said, it's pretty detailed, and next I'll show you. As I've said earlier on, January has been slow, which is usually the case. So for the first 2 weeks of January, I've been working from home. And so I've just been doing some disease models. So essentially, I'll create a look in our systems to see what fungal pathogen we have and then what agricultural crops those pathogens infect. And then I create a profile. So I call it, it's a disease model. And standard, you'll have, I'll show you in a few, where you have, you need the plant that is susceptible. So the plant must be prone to infection by the particular pathogen. The particular pathogen must be virulent, so it must be able to cause disease in the plant. And then you want the right environmental conditions for the disease to happen. So they need to meet under the right conditions for that to happen. And if it was in, say, medicine or human healthcare, then the human would be the host, and you might have your virus, your bacteria, or your fungal pathogen, and then the right conditions. So it's pretty similar in that sense when it comes to disease triangle. And then these are some of the things I've just been working on. I have some soybean, wheat, different wheat ones. And yeah, so remember I was saying that you need your susceptible host. So in this case, it would be your tomato. This would be the disease. The green mold is a disease, the conditions that you need. And then this is the pathogen. And we have different kinds of the same pathogens, but we'll have different numbers of them. And then the picture in the backdrop is just what the symptoms look like, because then we'll need to know what the symptoms look like so that we can assess for disease. So essentially, you'll grow up your pathogen and then you will have to put it onto the plant, wait for disease to show up, and then you sort of grade the disease. And after we've done that, then we'll try to treat it. So yeah, that's what I've been doing when I've been working from home since January. I'll be back in the lab next week, so yay! Here are two clips of what the pathogen system like. So that's rainfall being stimulated, and it is very misty, and it loves it. So yeah, see you next week. Bye.